Hello, everybody. Welcome to Science Abounds, Science at Home, where we will discover home lab magic. In this channel, we will be performing experiments demonstrating scientific principles, concepts, and reactions in chemistry, biology, microbiology, microscopy, and even molecular biology. Welcome to my channel. Hello, and again, welcome to Science Abounds, Science at Home, where we would discover home lab magic. Welcome back. Thank you for all my subscribers who have subscribed to my channel and hit the like button. It means so much to me. If you're new to the channel, welcome, and I hope you get a lot of fun and learning experience from my channel. And please, if you are new, go ahead and hit the subscribe button. It really does help me. In today's video, the experiment what we're going to be doing is we're actually going to be exploring gel electrophoresis. It's one of my favorite techniques in science, and I love using it. I've used it throughout my career as a clinical lab scientist, and it has many purposes and uses. And yes, you yourself, just like me, can do home electrophoresis in the comfort of your home with a very simple, inexpensive setup. And this is what I'm going to focus today. And I'm going to be running some DNA fragments of some bacterial pathogens just to illustrate how gel electrophoresis works and runs. And there's going to be a lot more videos in the future where I will be doing molecular biology and biotechnology experience, yes, from my home lab, using gel electrophoresis. We'll be doing DNA experiments, RNA. We'll even be doing polymerase chain reactions experiments in our home, and I will be showing you just how you can do that. So basically what I'm going to focus on is showing you the components of what makes up a gel electrophoresis system, and then in the second part I'm going to show you how to run some samples and look at the results. But this is not a molecular biology video. It's purely to explore and to demonstrate the technique of gel electrophoresis. I will be going into depth in future episodes, looking at DNA, RNA, molecular biology techniques and experiments. So please stay tuned for more exciting molecular biology, biotechnology videos coming in the future. So gel electrophoresis is a technique used to separate mixtures of DNA, RNA, and proteins according to their size, their charge, and to some other physical properties. It's widely, as I've already said, is widely used in biochemistry, molecular biology, and genetics to analyze samples. Electrophoresis works on the principle that charged particles move in an electric field. The gel used in electrophoresis serves as a medium to separate molecules based on their size and charge. When an electric current is applied, charged molecules move towards either the positive or the negative electrodes, depending on the charge. DNA is a negatively charged molecule, and so it migrates to the negative electrode. And it is negative due to the phosphate group in its backbone. So it will migrate towards the positive electrode. So I have my basic setup, and we're going to demonstrate how to make up the buffer, the gel, to cast the gel. The gel matrix that we're going to be using is made from agarose, and that is typically used for DNA studies and protein studies. It has small pores, and these small pores act like molecular sieves, and smaller molecules move through the gel matrix more easily and quickly, while larger molecules encounter more resistance and therefore move slower. And this is going to be very relevant in today's experiment. One final component that's very important in gel electrophoresis is the buffer. The gel is submerged in a buffer solution that maintains a stable pH and provides ions to carry the electric field. The buffer also prevents the gel from overheating during electrophoresis. We're going to be using TBE buffer. TBE buffer is commonly used buffer in molecular biology and is the predominant buffer that we'll be using for all of our gel electrophoresis experiments in the future. And it is in particular useful when we're separating DNA, RNA, and protein molecules. TBE stands for tris borate EDTA, which refers to the three compounds that make up the buffer. Tris acts as a buffering agent, maintaining the pH of the solution at around about pH 8, just above neutral. The borate helps stabilize the charge of DNA, and it also provides additional buffering capacity. And the EDTA chelates metal ions, which would otherwise interfere with enzymes like nucleases and prevent the degradation of nucleic acids. So that's what I'm actually going to do to begin with, is to make the buffer. And so you can buy these home lab electrophoresis system. They're very simple to use and to put together. And there are a couple of vendors or suppliers that I use or that you can purchase yourself an electrophoresis system. For one, Carolina Biological, and the other miniPCR. Both supply very simple, affordable electrophoresis systems. So please check those out if you're interested in performing electrophoresis experiments at home. Also, another resource is this book, The Illustrated Guide to Home Biology Experiments, published by Maker. And this book has a chapter on electrophoresis and how to build and construct your own gel electrophoresis apparatus and how to run a gel separation experiment. So please check them out. The details are in the description at the end of my video. So let's make up the buffer. So I normally make up the buffer in bulk. It's very stable. So depending on where you purchase The powder, you can get them in packets, 1-serve packet. So this whole packet is the amount that I need to make up 600 mLs of buffer solution. You can buy larger containers, depending on what you plan to do and how often you're going to do electrophoresis. You can buy big containers, small containers, or packets like this. You can get tablets, depending on the resources of the vendor of choice that you choose. But I list all my vendors at the end in my video description, giving you all the details where you can find everything. So I'm going to put the powder, the TBE buffer powder, into my container. Okay, don't need that again. And then what I'm going to do is I'm going to add 600 mLs of water. And when you're doing electrophoresis, you really want to use good quality water, distilled water. Don't use tap water. Okay. All right, I don't have a big enough container, so I'm going to make it up in a beaker and then find a better stoppered container in a minute. Okay, so I have a beaker now, a bigger beaker, so I just need to add the final 100 mLs, and then I should have 600 mLs of TBE buffer. I guess this container I thought was big enough, but it's not, and that's fine. Okay. All right. So I'm just going to let that dissolve. Give that a little mix. I'll get a glass rod and just mix that up a bit. Let that settle and mix. Okay, we'll come back to that in a minute. We don't need it right now. So let's take a closer look at the gel electrophoresis apparatus. Okay, so this is the main gel container or compartment where the gel is poured into and the whole electrophoresis system is put together and runs. So there's 2 electrodes that you use, a negative and a positive. So I'm just going to put these electrode like so at the back of opposite ends. And then I got 2 rubber rests, which I'm going to place my comb. And this is, the comb has different well numbers. And the comb makes your wells that are in the gel, which you apply your sample to. So I've got a 6 well comb and an 8 well comb. So for our experiment, we're just going to need an 8-well comb. So I'm going to put that in like so at the head of the positive electrode, the negative electrode, I should say. Okay. So the next thing I want to do is make my agarose. So I'm going to use a tablet. I got my agarose in tablet form, but they come in powdered form, in jars or whatever. So it depends whatever you use. I like using the tablet form. And then you dissolve that in 30 mLs of your TPE buffer, which I've got here, which I made earlier. And that's all been mixed and dissolved now. So I'm going to add 30 mLs of TPE buffer. Okay. And then I need to allow for the agarose tablet to disintegrate. The solution is going to remain cloudy at first, but we're going to heat that up to boiling. And we're going to boil the agarose so it becomes clear. So I do have my hot plate here ready. So I'm just going to make sure I just need the tablet to disintegrate. And it's very important that you use the TPE buffer to dissolve your agarose powder and not water. Okay, it's pretty much disintegrated. There's going to be a suspension in it. The solution does not clear until you bring it to a boil. So I'm going to place it on my hot plate, and it shouldn't take too long to boil. Okay, so it's boiling now. So I think it's probably ready and can be poured. As you can see, it's gone clear. Let me see if I can get some of the agarose that's on the side and get that dissolved. Okay, let me just bring it to the boil again. And then you immediately, when it starts to boil, you want to pour it into the tank. Okay. All right, it's nice and clear now. And what you want to do is you want to pour that into the gel chamber, like so. And just give it a little tap. You don't want to see any bubbles in the gel, if at all possible. All right. So you just leave that to harden, to solidify, and turn into a gel. Takes a good 20 minutes for it to solidify. So we'll come back when the gel is ready. Okay, so while the gel is setting and forming, let me go over the experiment and what samples we are going to be running. So I've created this scenario where we're working in a hospital microbiology department, and there is believed to be a suspected outbreak of a pathogen in the hospital's cafeteria. So samples have been collected from different areas of the kitchen and submitted for analysis in the microbiology lab. We've already performed PCR and created DNA fragments, and we're particularly looking at two potential pathogens in this scenario that they believe may be causing an outbreak. The first pathogen that we're going to test is Klebsiella aerogenes. Klebsiella aerogenes is a type of gram-negative bacterium, and we're going to be doing a whole video on gram testing of bacteria and learning about gram-negative and gram-positive bacteria. So stay tuned for future videos where we'll be extensively looking at the technique of gram staining, and you can learn a lot more about that. What a gram-negative and a gram-positive bacteria is, but that's beyond the scope of this video. So Klebsiella is a gram-negative bacterium. It is part of the Klebsiella genus. It's a common opportunistic pathogen that can cause infections, particularly in individuals with weakened immune systems. It is typically found in both the gastrointestinal and respiratory tracts of humans and can cause a variety of infections, including urinary tract infections, UTIs, pneumonia, and even wound infection. The other suspected pathogen is Staphylococcus epidermidis. This is a gram-positive coagulase negative bacterium that is a common part of the normal microbiota of human skin and mucous membranes. It can act as an opportunistic pathogen, particularly in hospital or healthcare settings. Common infections include infections associated with medical devices, endocarditis, and wound infection. So I've got two control samples. One is a Klebsiella control DNA sample, and the other is the Staphylococcus DNA. And then I've got 4 samples that were taken at different random areas of the kitchen of the cafeteria, and we're going to run those alongside the control samples and compare the banding. The Klebsiella will be a long DNA fragment and won't travel as far as the Staphylococcus, which is going to be a short DNA fragment, which will travel further. So any bands, and then what we will do is we will match up the bands against the controls to determine what infection or what pathogens we can detect. So that's the basic setup and the premise of the scenario that I'm using in order to demonstrate the technique of gel electrophoresis. Okay, so 20 minutes later, the gel has solidified and is now ready. I've already removed the electrodes. So all I want to do now is remove the comb. So you want to be very gentle and just lift up in a straight manner as much as you can. Okay, holding the gel chamber and just slowly lift up the comb. It's not easy, but okay, there we go. So the comb has been released, and as you can see, there are now 6 individual wells in the gel, and that's where we're going to apply the samples. So I'm going to remove these supports. We don't need those anymore. And then what I'm going to do now is I'm going to add 30 mLs of the TBE buffer to the chamber. So you want 30 mLs. Just enough to be above the wells, to cover the wells. Okay. And you just want to tap it, that you don't want any bubbles anywhere in the wells. Okay, so I'm going to insert the electrodes now. Like so. And the same with this electrode. Make sure. Okay. So now it's time to apply the samples. So like I said earlier, the PCR extracts have already been sent to the microbiology lab, and the PCR has already been done. We're going to save PCR to another video because that's not within the scope of this video. So I have the samples already provided, and these samples contain the dye already. We're going to talk a little bit about visualization and how you visualize the bands later on, but these samples already contain a dye in them. So what I'm going to do is I'm going to turn this around so you can see me apply the samples. And it's kind of difficult, so you do have to practice, and if you don't get it right the first time round, that's perfectly fine. We don't expect you to get it right the first time round. So the first two samples are going to be the markers. Okay, so I've got a pipette and I've got some pipette tips, and I've got some more pipette tips with me, just because I will need more. And what you want to do is, when you're putting your tip in, you want to place it just above the well. And what I'm going to do is I'm going to go backwards. I'm going to apply the first to the nearest well to me, the control. So the first two rows will be controls and the next will be my sample. So I'm going to take up some of the sample, like so, sample 1. And then what I'm going to do is I'm going to hold the tip and I'm going to place it just below the surface of the buffer, and then press in, and it will sink down. It doesn't matter if some of it spills over into the buffer, that's fine. So that, again, the technique, you just need to practice. If it doesn't look very nice the first time round, you just keep going. but that's my first control. Next is my second control. Again, just below the level of the buffer, holding the tip in place to keep it steady. And it will sink down into the well like it's doing now. Okay. So now I'm going to do the first of the sample. So sample A was collected from the meat preparation table of the kitchen. Okay. Again, it doesn't matter if anything spills into the buffer surrounding, it doesn't matter. It's not going to interfere with the electrophoresis. Sample 2 or sample B is going to be from the serving table of the kitchen. This was collected from the serving table of the kitchen. Okay. And what is funny is that a lot of people, including myself, hold their breath when they're applying the sample to the wells. It's very easy to do, just to hold your breath. But if you find yourself holding your breath, that's fine. Sample C was collected from the washing up area of the kitchen. Okay. And then the final sample to be applied, got another tip here, was taken from the vegetable cutting table. Okay, so I'm going to pipette the final sample and apply that to the last well. Remember, we used a 6-comb template. Okay, lovely. So now I've applied the samples. So now what I'm going to do is I'm going to attach the electro wire to these crocodile clips. And then I'm going to attach it to the power source, which I have here. And it's going to take about half an hour, 10 to 15 minutes for the electrophoresis to take place. So we just let that run now. Okay, the electrophoresis is complete, and we've got separation of different bands. I misspoke earlier about the DNA lengths, the short length and the long length. The short DNA fragment belongs to Klebsiella, and the long DNA segment belongs to the Staphylococcus. So I'm going to stop the electrophoresis experiment and unhook my electrodes. And we're going to take a look at our results. So if you remember, I went from right to left. Normally, when you're loading a gel, you do left to right. But for the demonstration of today's experiment, it was easy for me to go from right to left. But you do it whichever way you want. Just remember what you're putting in each well. So the first well was my Staphylococcus DNA fragment control. Okay, as you can see, it's only one band and hasn't moved very far from the actual loading well. So it's a lot, that shows you that it's a long DNA fragment and moves slowly in the gel. The second control, I have two bands. Okay, in the second well, I have two bands. So what I've done here is the control I added to the Staphylococcus control the Klebsiella. So the first band is the Staphylococcus control, and then the second band is the short DNA fragment because it has traveled longer than the first band, and this is my Klebsiella control. So well 1 is Staphylococcus, well 2 is Staphylococcus and Klebsiella. In well 3, remember, sample A was taken from the meat preparation table and has two bands, and that matches the control with the Staphylococcus and Klebsiella. So we know we have pathogen detected in the meat preparation table. In the next well, sample B was from the serving table, and here has a single band which matches the Staphylococcus control. So this sample is positive for Staphylococcus. In well 5, sample C was taken up from the washing up area, and again, a single band that matches the Staphylococcus control. So the sample was positive for that, for Staphylococcus in the washing up area. And the final well 6, was sample D, which was the vegetable cutting table. And here we have two bands representing Staphylococcus and Klebsiella being detected. So basically, the kitchen has a serious contamination problem, and the microbiology department reported back to the cafeteria manager, and a major clean down and decontamination process was instigated. The experiment was repeated after the cleanup and disinfection, and all results collected were negative. So they were happy that they contained the outbreak and were working in a sanitized area. So this is just one example of how gel electrophoresis can be used. And we're going to be exploring many other uses and experiments that gel electrophoresis can be used. And it's a very exciting technique. So just a little bit of information about visualization of the bands. So after electrophoresis, the separated molecules have to be obviously visualized. And for DNA and RNA, the gel is usually stained with a dye called ethidium bromide that binds to the nucleic acid. And under UV light, the dye fluoresces. And we're going to be using that dye sometime in the future. Also allowing visualization of bands for proteins, we often use the stain Kumasi Brilliant Blue, and this can be used to visualize the bands as the gel progresses without any UV light or any special lighting. So today our samples contained a dye that we were able to visualize without the aid of any special lighting. So that's just one example of how we can visualize the bands, gel electrophoresis bands. Well, I hope you enjoyed that video, that experiment of gel electrophoresis and how important it is in the world of molecular biology, biochemistry, clinical laboratory sciences, and genetics. Please hit the subscribe button. It's totally free and it helps support my channel, makes me keep exploring science and developing more and more experiments. Follow along with me at home by my procedures that I publish on my Patreon page. You can get all those and also exclusive experiments that aren't shown on YouTube can be seen on my Patreon page for those who become members. Please email me, send me your questions, concerns, photos, and videos of you doing home lab experiments. I want to build up a home lab community. Please do leave me a comment. I always love to hear from you. Until next time, keep on experimenting. Goodbye, everybody.